Nice Mars map.
Hang on a sec. Okay, so here's what we're going to do today. We're going to go over a few different topics in high resolution microscopy. Then I'm going to show you something. I'm still in the process of writing the final exam, but I'll show you something relevant to the final exam, which will be given out Thursday morning, and you'll have until the following Friday, the 15th, to work out the two projects. I want to talk first about confocal and nonlinear and multiphoton microscopy. So, basic fluorescence microscope. We've got something in the ground state. It absorbs a high energy photon. It emits a lower energy photon because it loses some energy to vibrational transitions. And of course, the problem with this is that you're shining light onto a specimen that's been set up to glow. And so everything inside your specimen could potentially be glowing. Anything between the objective of the lens, the objective and the part of the sample you want to look at could be glowing. So what they do is they set up a pinhole. And the basic idea is you've got an objective, and there should be a beam splitter somewhere in here so that the excitation light would be coming up like this, hits the beam splitter, reflects that way. And you can set up a beam splitter so that instead of being 50-50, it's very high reflectivity at the laser wavelength and very low reflectivity at other wavelengths. So, comes in, gets focused. Specimen glows. It's been labeled with something that will make it glow. The emission comes, gets collimated, passes through the beam splitter because the beam splitter has a wavelength-dependent reflectance. Then another lens focuses it onto a pinhole. And if it's coming from the focal point, of course, it goes through the pinhole, no problem. But if it's coming from somewhere else, well, it doesn't get focused onto the pinhole. Now, that doesn't mean that there are no rays who go through the pinhole. Of course, there's always a few rays that go through the pinhole, but there are way more rays that go somewhere else. And so you can reject most of the background using this pinhole detection. Now, of course, that means that you have to scan. It means that you have to go point by point. So you're not taking a picture of the entire specimen at once. You have to scan the light. You could do it by moving the specimen. You could do it by moving the lenses and detectors, or you could do it by playing with some mirrors so that you change where this is focused, and you send the light in at different angles, and then you also play with the mirrors back here in some way to undo that deflection angle so everything keeps going to the detector. But that's the basic idea. Any questions? All right.
So even if you're
What?
So even if you're blocking all of that out of focus light, you're like, what's the best resolution you can get with a microscope?
I mean, the best resolution you could get with a normal confocal microscope would still be diffraction limited resolution.
Right.
What you're getting out of it is that you're getting background rejection. If there's a whole bunch of stuff in between the focal point and the lens that's glowing, if you're looking at a thick sample, you've got a bunch of stuff that's fluorescently labeled. So there's glowing stuff here. There's glowing stuff there. And all of that other glowing stuff would also be sending out of focus light to a camera. So if we didn't have the pinhole, if we just had a camera, and we didn't have the second lens, if we just had a regular camera, so we have the objective, we have a tube lens, and we have a camera, then we would get all of the focused image from this plane, and a whole bunch of out of focus light from these planes and these planes. And if your sample's pretty sparse, maybe there isn't much out of focus light. But if your sample is not sparse, if there's stuff everywhere, then you're getting all this out of focus light superimposed on top of your image. That's what you're getting.
Gotcha, that makes sense. Thank you.
You're welcome. All right, so here's a more detailed version of how it might work. You've got your specimen, you've got your objective lenses, and there are two mirrors. And the idea of the mirrors is, well, if we've got parallel rays coming in, they focus a distance y equals f tan theta from the focal point. So I could play with these two mirrors to move the focal point in 2 dimensions, and thereby look here or here or here or wherever. And then what I could do is the light, of course, comes from that point. I've got a point source that's off axis because I've just focused the light here. I've made a point in the specimen glow. That glowing point sends its light off axis. Well, then these two mirrors undo all of that. And when these two mirrors undo all of that, just like the laser light was coming in on axis here before getting deflected by these two mirrors, that deflection gets undone. So it winds up going in a straight line on axis again, and it hits a beam splitter, and it's a beam splitter that is very high reflectivity for the laser light and very low reflectivity for the emission light. So it gets focused to the pinhole. Whatever makes it through the pinhole goes to what's called a photomultiplier tube, which is basically, for some of these things they use it's not a photo avalanche it's not an avalanche photodiode, but you could think of it as being like an avalanche. Avalanche photodiode is a technical term for a very specific type of light detector. But basically in a photomultiplier tube, light comes in, hits something, and ejects an electron. That electron that gets ejected is then accelerated so that it slams into something else and knocks off more than one electron. All of those electrons are also placed inside an electric field. There's a high voltage difference which accelerates those electrons, which creates yet another burst of electrons. And so basically one electron gets multiplied into many. And the reason why they use this multiplication process is that it provides great discrimination between 0 and 1. If you only get one electron, well, that's a very, very weak signal. This takes a single photon and multiplies it. Now, the precise number of electrons that you get is highly variable. So 2 photons at once, you're not going to be able to necessarily tell accurately. Maybe you could tell 2 from 1, but telling 3 from 2 would get harder, and telling 4 from 3 would get harder still. So basically, you have a detector that every time there's a photon, it gets a little spike. And you just count the number of spikes, and that tells you how many photons you get. That's what a photomultiplier tube is.
So it's detecting the current in a sense.
Yeah, you're counting the spikes of current.
Right, okay.
And exactly how big that spike is, well, it's a random multiplication process. One electron gets turned into many. There's no guarantee that each conversion will be 100% efficient, which means that each multiplication is random, but it'll be a spike that's big enough to notice. And so with these photomultiplier tubes, you just count the number of spikes that you get in some time interval. And then you have to count on the fact that the spikes will not come too close to each other. So you have to set up an experiment where that's a reasonable proposition. Often that's a reasonable proposition. How close is too close? Well, it depends on how long the spike of current lasts in here, which is determined by the electronics. but all of these things are feasible. So you can do 3D sectioning. Here's somebody got a bunch of PMMA, polymethyl methacrylate. It's basically plexiglass. It's related to plexiglass. A bunch of little plastic spheres. And you can make plastic spheres that have fluorescent dye incorporated into them. And then they just put those into some sort of gel so that they're frozen in place. They have a fluorescent dye so they'll glow when you shine light on them. And then you can image them layer by layer and reconstruct a 3D map of their positions. And of course, you could do that with more complicated things than these spheres, but that just illustrates the concept. Now, the problem is photobleaching. If you linger too long, then eventually these dyes will get damaged. And you could say, well, I'm not focusing on any one region too long. No, you're not focusing on any one region too long, but the light has to keep traveling through that region to get to the layer below. And so everything is getting exposed to a lot of light. And depending on the dye you're working with, the amount of time you're looking at your specimen, that may or may not be a problem. Here's something on how you have to optimize the size of the pinhole. Basically, if it's too small, then if you look at the focused light on the detector, it's blurred. We'll skip that. So then we get to two-photon emission. And the beauty of two-photon emission is that unless two photons arrive at the same time, the thing won't end up in the excited state. Now that matters for the bleaching because bleaching preferentially happens in the excited state. Bleaching is basically an irreversible chemical reaction that damages the molecule, damages it in such a way that it no longer glows. You change one of those bonds, which changes the electronic structure, it's no longer absorbing and re-emitting. And those types of reactions are more likely to happen in the excited state. So if we could limit the amount of time that something spends in the excited state, if we could make it so that these things, when they're out of focus, don't absorb any light, then we can drastically reduce photobleaching. And the beauty is that whereas with one photon microscopy, anything that the photon encounters is fair game. Any place in the specimen where light travels while getting to the focal point or getting past the focal point, if it didn't happen to get absorbed there, fair game, it could absorb. And while it's obviously more likely to absorb at the focal point because there's more photons at the focal point, there's also plenty of exposure at other times. But with two photon, really, unless you're right at that focal point, the odds of two photons being there at the same time are really, really low. So you absorb two photons, neither of which has enough energy to raise you to the excited state. You emit one. So it's automatically confocal. And I want to switch files to go through that calculation. So why is the view not changing? Okay, now I should be able to do it. View, full screen mode. All right, so let's look at, here's a lens, here's rays of light focused, and here's a little segment of the specimen where the light is out of focus, hasn't yet reached the focal plane. This segment has a width W. It has a thickness dc. And we can ask, how much light do we get from this? Well, the power per molecule will be the intensity of light hitting the molecule times an absorption cross-section times the fluorescence efficiency. And that fluorescence efficiency is basically what is the probability that something in the excited state will lose its energy by emitting a photon. The molecule in a vacuum generally there are very few ways to lose energy. But if it's in a solution, there are other ways to lose energy. It's constantly being buffeted by water molecules and other things. There are other ways to give up energy from that excited state. Well, that intensity is power over pi times the beam radius squared. Now, let's ask how many molecules are excited in this volume. Well, that's going to be the volume multiplied by the number density, number of fluorescent molecules per unit volume. So we've got a pi w squared dz. When we multiply the intensity per molecule by number of molecules, the pi w squared up here cancels out the pi w squared down there, and we're left with something constant. We're getting a constant amount of light from each plane. And just as we're getting a constant number of emission events from each plane, we're also getting a constant number of bleaching events. Now, of course, here the bleaching events are spread out over a wide area. Here they're spread out over a smaller area. Here they're spread out over the smallest possible area. But still, the point is that the number of bleaching events within this area is constant. And there will, if I'm looking at this region of the specimen, we're getting an intermediate number of bleaching events. If I later change my scanning, so now we're looking at this plane, well, I'm going to get some bleaching events again in this volume, concentrated into a smaller portion of it. And everything in the course of the experiment winds up getting the same number of exposures. So it's a really bad idea. But what if we have 2 photon or 3 photon absorption? Then the power per molecule is proportional to the intensity to the power n, where n is the number of photons that have to be absorbed at once. Because basically, it's the probability of having 2 photons or 3 photons, or nowadays people can even do 4 photon in the same place. I mean, if you were to think about what's the probability of any one person in a room having their birthday today, it's roughly 1 out of 365, ignoring the fact that birth rates aren't constant throughout the year. What's the probability of 2 people having birthdays today? Well, it's going to be 1 out of 365 times 1 out of 365 times the probability that no one else in the room does. But you could ask, what's the probability of having at least 2? It's going to be proportional to 1 out of 365 times 1 out of 365, which is the square of the probability of 1 person having their birthday today. So likewise here, if we think of the intensity as being proportional to the probability that you get a photon at any given instant, then the probability of getting 2 has got to be proportional to intensity squared. So we've got, when we square the intensity, power to the n over beam radius to the 2n. Because we squared, we take w squared and raise it to the power n. And then we multiply by pi w squared, and we get 1 over w to the 2n minus 2, or for 2 photon, 1 over w squared, and 3 photon, 1 over w to the 4th. Which, if we look at these 2 figures, explains why the fluorescence is very bright near the focal spot in 2 photon and very spread out in 1 photon. Here's an experimental illustration of that. They illuminated this sample of dye. This is just some fluorescent dye dissolved in liquid. And here they hit it with a laser that produces 1 photon absorption. Here they hit it with a laser that produces 2 photon absorption. And you can see the difference. Any questions? Now this is good for more than just eliminating photobleaching. Besides eliminating photobleaching, it also really, really reduces out of focus emission. Here, you can see that there's going to be lots of out of focus emission. Down here, there's no out of focus emission. You might be saying, well, this isn't a problem. We can just do confocal. We'll just set up that pinhole. All right, we could set up the pinhole. But what if we're getting very few photons? Then the only photons, if very few photons are being emitted, then the only ones that we're going to collect, all those ones coming at us from the side right now, those aren't going to go to the objective here. But if we don't need the pinhole, if we know that all of our light can only be coming from here, then we could surround this thing with detectors. And in fact, I know people who have done experiments like that, where they have surrounded the thing with elliptical mirrors so that they collect light coming from all directions to make the maximum possible use of their light. Now, you still can't surround it in every possible direction, but the point is that you can cover a wider range than if you just collected through the microscope objective here. You could at least cover some additional sides. You can multiply your light collection easily by a factor of 4. 4 times as many photons is nothing to sneeze at. And this is why people like to use multiphoton microscopy in, say, neuroscience, because brain tissue is inherently cloudy. It's not completely cloudy, or you couldn't see through it and do these experiments at all, but it's very cloudy. And so it's hard to get a lot of light. So basically, any photon that makes it to the surface of the brain, when they're looking at a mouse brain, they want to take that photon. No matter what direction it's going when it comes out of the brain, they want to take that photon. So they don't want to just look through the excitation optics and then set up a pinhole. They want to collect light from multiple angles coming off the surface of the mouse brain. Any questions? All right, then let's talk about a different kind of microscopy that people do when they've got highly focused light that's been intense and pulsed. First, you excite the light, right? First, you excite it. Sorry, first you excite the fluorescent molecules. Then you hit it with a second beam. And the second beam is called a donut mode because it has 0 intensity in the center. There's a ring of light and there's 0 intensity in the center. And the way you set up a donut mode is basically this. Instead of just having your objective, you put a filter in front of it. Now, I've drawn this part dark, not because it actually blocks the beam, but to indicate that half of this filter induces a half-cycle phase shift. Maybe half of this has a slightly different refractive index, so that the light traveling through this part of the filter takes an extra half cycle to get through. Which means that instead of all of the paths taking the same amount of time, half the paths take the same amount of time. The other half the paths take that amount of time plus a half cycle, which means that this has gone from being a point of constructive interference to being a point of destructive interference. Any questions on that idea? Now, the reason you want to do that is that anything that's right in the center has just been hit with a second beam of light. And that second beam of light has its wavelength tuned to this downward transition, all right? Tuned to whatever the strongest downward transition would be. And if it's tuned to that strong downward transition, remember how last week we talked about stimulated emission? We said that lasers work on the principle of stimulated emission. You've got a molecule hanging out in its excited state. It's going to emit a photon at some point. But then another photon comes around that's at the same wavelength and frequency as the one that was going to emit, and it gets it to emit sooner. This is basically a resonance condition. I mean, you think about a tuning fork. You have a tuning fork reverberating. It's producing sound. And if the sound is at the frequency that a piano string could produce, then it will start resonating with it. It's not quite the same idea because the piano string is not primed in an excited state waiting to go, but it's as close of an analogy as I'm going to get in classical physics. So you hit it, you've got something hanging out here in the excited state. And if we just leave it to its own devices, it's going to be there for nanoseconds. But if I don't wait nanoseconds, if I just excite and then a few picoseconds later hit it with something tuned to this downward transition, all of this stuff out here is going to make that downward transition. What's left right here? Well, this stuff can't make the downward transition. Anything that's hanging out here is still stuck in its excited state. And so a few nanoseconds later, which doesn't sound like much time, but to a molecule, that's an eternity. A few nanoseconds later, it emits. And so what we know is that any light that comes out a few nanoseconds later must come from this very tiny region. Whereas any light that came out just a few picoseconds after first raising stuff to the excited state, that must have come from out here. And so what we've done is we've now set it up so that we can discriminate, we can tell the difference between what's coming from out here and what's coming from in here. Even though we had diffraction limited light focused down to a spot size 0.6 lambda over the numerical aperture, we've just set it up so that our emission is coming from something much smaller than 0.6 lambda over the numerical aperture. All we have to do is make sure that we have 2 pulse lasers, exquisite timing control, a filter that will take half the light and retard it by a half cycle, and detection electronics that can discriminate on nanosecond time scales. That's all we need. You know, nothing too expensive. Just, you know, a few $100,000. Back in the day, probably $1 million. Nowadays, a few $100,000. Any questions?
What's the most expensive component?
Not too long ago, I would have said the pulsed lasers, but those things, they keep finding ways to improve those. So I'm not sure whether it would be the pulsed lasers or the microscope. The microscope objective itself would probably only be $10,000, $20,000. But getting that microscope objective as part of a well-aligned, mechanically stable thing, that takes you into the $100,000 range. I'm not sure.
I have another question.
Yeah.
So you can discriminate between the fluorescence transition and the STED transition.
Yeah.
Time interval, but like in addition to the time interval, they also have like different wavelengths. Like giving out slightly different.
There is okay. The stimulated emission, the STED emission, will be at a very specific wavelength. This fluorescence that happens later will be over a range of wavelengths. The problem is that this range of wavelengths will still be pretty narrow, still be pretty close to this one. Anything that could just block this would also block a non-trivial fraction of this.
I see.
So I don't think people normally do that. I mean, in principle, they could. I know I am a casual acquaintance with the guy who invented this stuff. I'm sure he's played with that. Cool. I guarantee you he's played with that. Other questions? All right, so here's just some examples of increased resolution, conventional confocal microscopy. You see a bunch of blurs, stimulated emission depletion. This is from 2006. I borrowed someone else's lecture notes. Everything has gotten way better since then. What else do I want to say about it? I mean, this is just basically an illustration of how you narrow things down. I could add that because the beam profile here is proportional to the squared distance from the center of the beam. It's 0 here and increases quadratically out here. Once r squared times a bunch of constants reaches like a threshold, where you now are sending an appreciable number of things to the ground state, that's going to determine the size of this beam. So r squared times your intensity has to equal a threshold, or r is inversely proportional to the square root of the intensity. And so the more you can turn up this beam, if you turn up this beam really, 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 really high, then you could, in principle, get this down to single molecule resolution. But all of that is contingent on the idea that all of these paths really did take the same amount of time, so that this filter is really not, so that this filter is producing truly destructive interference or truly zero intensity there. And so this can actually become aberration limited imaging again, even though it seems like this is beyond diffraction limited. In fact, this is aberration limited because we're counting on truly zero intensity there. We're counting on the idea that you could just keep cranking up the intensity. And if you crank up the intensity enough, this quadratic profile could be above a threshold where everything gets sent to the ground state once you're more than 1 molecule off center. You just keep cranking up this intensity, and even just a little over 1 molecule off center, you are at the threshold where essentially everything's getting sent to ground. But what if this isn't quite 0? Then as we crank up that intensity, this molecule could also be sent to ground. So this is actually aberration limited imaging again. People have spent decades playing with pulsed lasers and time-gated detection to beat the diffraction limit, only to find themselves staring at aberration limited imaging again. Any questions? Here's some more pretty pictures. That's one of the beauties of this field. You produce lots and lots of pretty pictures. I want to talk to you about another way of beating the diffraction limit, what I have spent most of my time on for the past decade. And I'm a theorist, so why am I working on this? Well, this is from the Zeiss company website. Zeiss is an optics company that's around to this day. In the 1860s, Ernst Abbe, the guy who came up with the theory of the diffraction limit, was a professor and also a member of the scientific staff at Zeiss. Much like some of today's top professors, he split his time between academic work and consulting for industry. The world doesn't actually change. And what they said about Abbe is that before his work, microscopes were built on a trial and error basis. After that, they had a benchmark. They knew exactly what target to aim for. They knew that if they were close to that target, then any further progress would be incremental and they could invest accordingly. They knew that if they were far from that target, there was still a lot of room for fundamental progress, and again, they could invest accordingly. So they could calibrate their time and resource investments to how much potential gain there was. And that's why I work on this stuff. Because again, people have come up with a new technology, so we need a new benchmark to aim for. So if we had an ideal lens, of course, 2-point sources would just make 2 small point images. Instead, we get a blur. We've talked about what that's from. I'm going to skip some of that. But here's something we haven't talked about before. There is still sub-wavelength information in here. All right? This is a simulated image. And we're going to say that the molecule is perfectly centered on one of the pixels, or perfectly, it's at a point which when image corresponds to the center of a pixel. So the image is symmetric. Then we shifted a quarter pixel. And this side is a little bit brighter than that side. And this is a little bit brighter than that. So to the eye, even though this spot is roughly a wavelength wide, we can see a subwavelength shift. That's the information that we can work off of. And here, it's been shifted a half pixel, so instead of being at the center of a pixel, it's on the border between two pixels. Everything is symmetric here and here, here and here. So the idea that some people have worked on, and people have won Nobel Prizes for, is that instead of looking at two molecules at once, what if the molecules are blinking? So only one of them is shining, and later the other is shining. And with each one, you look at this bright spot, you find the center of the bright spot, and you say, at the center of this bright spot, there is a molecule. At the center of this other bright spot, there is another molecule. And we build up a quantitatively valid map of the molecule positions. So the basic sequence of events is this. We've got a whole bunch of fluorescent molecules. Only a subset of them are emitting light. And they emit a whole bunch of photons, so we could draw a circle corresponding to the image of one of them and find the center of that circle. Later on, a different subset are emitting light. Again, those images are circular. We can draw a little dot at the center of the circle. Occasionally, there are overlaps, and these overlaps are a big problem that I've worked on a lot over the past decade. Now, what's limiting you is, remember last week we talked about shot noise. We talked about the fact that the number of photons you get fluctuates, and they come one at a time in an unpredictable sequence. So if we just get 50 photons, well, your guess is as good as mine where that molecule is. As we get more photons, we get a more precise image. And it turns out that the accuracy with which you can estimate the position of that molecule is the wavelength of light over the square root of the number of photons detected. And the basic scheme is something like this. You've got a default state, which we will call the dark state. It's a state that isn't absorbing light from this beam and then re-emitting. Maybe it's absorbing, but if it is, it isn't re-emitting. You're not getting fluorescence if something is in the dark state and you hit it with this beam. But if we hit it with just the right kind of light, we can take it to a metastable active state. Metastable meaning it's partially stable. It's going to be there for a while, but not forever. And then this active state, well, if we hit it with the green photon, it absorbs, loses some energy, re-emits red. Goes back to here, not to the dark state, goes to here, hangs out here a while. So it can absorb another green photon, re-emit red. Absorb another green photon, re-emit red. And you could easily make this cycle go on for microseconds. And if the lifetime is nanoseconds, then in microseconds, you could potentially absorb and re-emit hundreds of photons. So you could get an image that has hundreds of photons in it, and you could determine the center of that image to a precision that's wavelength of light over square root of a few hundred, so lambda over 20. That's way, way, way beyond the diffraction limit. Now, eventually it will return to this dark state, but that's okay. You got an image with a few hundred photons. And you can set the number of molecules that go from the dark state to this metastable state by tuning the wavelength, by tuning the power of another laser, all right? The laser that sends things from here to here, well, what if that's a really, really weak pulse? So weak that only a tiny fraction of the molecules absorb it and go from the dark state to an active state. Well, that just means that we've got one of these scenarios here, where only a fraction of them are in that active state. Now, it turns out that people have apparently known for a while that there were molecules that would blink on and off, that would not just steadily absorb and re-emit, but would spend a lot of time in dark states. And basically in the mid-2000s, around 2000, depending on who you ask, 2004 to 2006 to even 1992, depending on who you ask, eventually someone said, that's not a bug, it's a feature. It wasn't until 2006 that a whole bunch of people together fully processed what that feature is and what they could do with it. And then a revolution happened. And in 2014, a Nobel Prize was awarded for work that, frankly, 4 or 5 groups deserved credit for, but only 3 could get credit for because of the rules of the Nobel Prize. More pictures. These are diffraction limited. These are super resolution images. Here you can see the details of individual microtubules. Here you can see microtubules, and you can also see the mitochondrial membrane. And they did the labeling with 2 different dyes, and they're playing with 2 different wavelengths, 2 different cameras. So they've got beam splitters and filters set up. This is not a cheap or easy experiment to do, but the result is that you can simultaneously look at the cytoskeleton and also the mitochondrial membrane and actually see inside. It's amazing that we can do this. These are some pictures from Eric Betzig's Nobel Prize literature. The main thing to take away is just how good the resolution is. This is from Xiaowei Zhuang's group. Personally, I think she should have gotten a Nobel Prize for this, but whatever. There's a whole bunch of things messed up with the 2014 prize. These are proteins in the axon, which is a little thing that comes off of the neuron. And you can see that periodic ordering of the proteins in the axon at spacings of, roughly 180 nanometers. And not only can you see the 180 nanometers, but you can very clearly see the gaps between them. And I just find it amazing that we can look at the molecular machinery of life like this. Because when I was in college, they told me that this is impossible. And stupid me, I believed them. There were other things I didn't believe that I should have believed, but I believed them when they said this. And then lo and behold, about 7 years after I graduated from college, I saw people looking with resolution better than this. And then in 8 years after I graduated from college, people just completely shattered that barrier and started doing things like this. And so I'm very pleased to report that something that I told you this semester is a lie, but I'll be damned if I know which thing was a lie. I wish I knew, then I'd work on that and get a Nobel Prize. But something that we told you in college is a lie. The problem is we don't know which thing. All right, now there are actually many ways to look at small things inside a cell, but this can be done in live cells, unlike transmission electron microscopes. It's non-invasive, unlike the near-field microscopes, which basically stick tiny little fiber probes on the surface of a cell. It's not limited to pairs of probes, unlike something called FRET, Forster Resonance Energy Transfer. It's a much simpler setup than stimulated emission depletion. You do need a very good camera, and those cameras keep getting better. But you know, that's still, it's expensive, but they're getting more affordable, and they're certainly easier to set up than all of the multiple lasers and filters and everything, and time-gated detection setups that you need in stimulated emission depletion. There's less bleaching than in some materials, and for those of you who have heard of negative index lenses, I guess what I would say is that yes, negative index lenses can in principle smash right through the diffraction barrier, but these materials actually exist, so that's nice. You can use it to test hypotheses like look at the structuring of, look at the clustering of molecules. You might just look at this and say, well, they're randomly distributed. Actually, they're not. People have done simulations where they guess at different distributions of spacings. And you could do statistical analysis on the spacings here and statistical analysis on the spacings in the simulation and see if the distributions match up. And if the statistical profiles match up, great. If the statistical profiles don't match up, that means that there's something about the way that the molecules interact in order with respect to each other. A big question, though, that I've worked on is how do we know whether there's one or two molecules in an image like this? So we came up with a model of, you've got a diffraction limited spot, size roughly 1 wavelength, and the one lever you get to turn is the probability that any individual molecule will be excited. And that's set by the activation pulse power. We could calculate the probability that you get exactly 1 activated in this spot. Because if you get 2 activated, they're going to have overlapping images. And you maximize the probability of getting 1 activated if the activation probability is 1 over n. Now that's not too surprising, except that we could calculate the probability that you got 2, 3, 4, 5, all the way up to n molecules activated. And we could compute the ratio of that probability to the probability of exactly 1 being activated. And what you get is 0.7, which means that you're getting, for every 10 single molecule images you also get 7 multi-molecule images. That's not good. So there's 2 ways that you could deal with this. You could activate fewer molecules, but that's going to mean a slower experiment. Or you could toss out the overlap data. That's going to mean a faster experiment. And so to toss it out, you need to feed the data to an algorithm that can guess whether or not there's 1 or more molecules in here. And everybody's come up with different algorithms for trying that task. What matters is the fraction of the M molecule images accepted. You want F1, the fraction of single molecule images, to be high. You want F2, F3, F4, etc. to be low. So we can recalculate our error rate. And we get that our maximum error rate depends on the algorithm performance, which sort of makes sense because this is a concept of microscopy where you're limited by how well you can analyze images and find their centers. So your error rates should depend on algorithm performance. And we can calculate the minimum number of activations or the minimum number of data frames you would have to take versus your target error rate. And we get a complicated formula. The main thing to know about this formula is that we can actually predict the relationship. We can predict the relationship between the number of data frames you need to collect or the duration of your experiment, the performance of your data analysis algorithm, and the error rate that you're looking for. And people can design experiments around this. Any questions?
So F1 and F2 are known right now.
F1 and F2 have to be calculated for each data analysis algorithm. So what you would basically do is feed a bunch of images, either computer simulated images or real images, but they have to be images where you know whether you have 1 molecule or 2. You feed those to your algorithm, you feed those to your data analysis software, and you see what does my data analysis software give. And then you know what F1 and F2 are. You know what fraction of those are accepted. And I do stuff like this with a friend of mine at City of Hope. I come up with computer simulated images and I feed them to her data analysis software. And we're answering different questions. This calibration has already been done a long time ago. I'll get to some of those later, but basically we're asking questions about how do we think the molecules are distributed? And if we think the molecules are following this pattern or that pattern, then I write a program that generates data based on that generates data that should be what her microscope would produce. It should look like what her microscope produces. and then we feed it to her image analysis software. Here's the fun part. We can look at higher order rejection algorithms. So we could say, hey, what if we've got something that can solve the inverse problem for 2 molecules or 3 molecules, but not for 5? And of course, in reality, that thing would be able to solve some 2-molecule cases, but not all 2-molecule cases. Eventually, they'd be so close together, it can't solve it. It could solve some 3 or 4, and there might even be a few extreme cases of 5. We're making an idealization here. And we're going to assume that this thing works for up to whatever number of molecules, and if it gets more than that, it malfunctions, but it knows that it malfunctions, so it tosses out that data. And we can calculate the number of data frames that you would need to collect for algorithms at different levels of intelligence and say, all right, if you can analyze up to 10 molecules, then the number of data frames that you need, it's not actually 0.06 frames, but it's 0.06 multiplied by a whole bunch of other constants. And so we can predict a minimum experimental time for an algorithm of very high intelligence. And that was a lot of fun to predict. And so far, all of the experiments have been slower than our predictions, which makes sense because we did an idealized bound. We predicted a lower bound on time, not an upper bound on time. Just give you a quick tour of some other things that we do. I want to give you a flavor of why someone who's theoretically inclined would choose to work on stuff like this. Statisticians have known for a while about something called maximum likelihood analysis. Basically, you've got your image, and you've got some model for the noise in the image. You've got some mathematical model that says, all right, if we've got a molecule that's here, I'll get on average however many photons plus or minus some range. That's the idea of the noise. And then we try to calculate the probability of getting different levels of noise given that the molecule is here. Now, if the molecule is here, well, then this data is highly probable. If the molecule's over here, it would require a whole lot of flukes to get high counts over here and low counts over here. And of course, this is an extreme, but choosing between here and here, well, you know, we'd have to calculate the probability of different flukes, and we can do that. And that turns out to be the most accurate way of doing this kind of analysis. But what we realized is that since we can model these point spread functions as products of Gaussians, this is a separable problem. We can turn this into 2 problems, one for X, one for Y, by summing the data in columns and then summing the data in rows. And so instead of doing L squared data points, L wide, L tall, we could work with 2L data points and speed this up, which is an important concept in computer science. I won't bore you with the details of the test. I'll just say that here's the fundamental limit. Our algorithm in every case that we tested it on was worse than the fundamental limit, but not too much worse. This is 3.5 here, and generally we were around 5. So we were roughly 1.5 times the fundamental limit in very non-ideal cases, which is not bad. Then we simulated a whole bunch of images centered here and here and here and here and so forth. And for each set of images, we calculated the mean and we calculated the standard deviation. And we said that the radius of these circles that we drew was twice the standard deviation. The key thing you should take away is that the algorithm isn't biased. Everything stacks up in a row as expected. And also that the radius declines as 1 over the square root of the number of photons. So every time we multiply the photons by 4, the radius shrinks by 2, which is exactly what theory would predict. We did some stuff where we played around with bleaching. We said, hey, what's the optimal acquisition time if, as the experiment goes on, some of the molecules go away. So initially you have a whole bunch of things here. Later you have fewer things in your image, which means that you can activate a higher fraction and not have to worry. You know, if you've got 20 molecules, you can only activate 5% of them before you get problems. If you have 10 molecules, you can activate 10% before you have problems. So you can speed up your acquisition. So you have to model the population versus time, taking into account the rate of bleaching. So you have to calculate the number of activated molecules, which depends on the activation probability, which is related to an error rate and algorithm performance. And then we can calculate the rate of bleaching, which depends on, again, error rates and algorithm performance and some parameter of the molecules. And so we could actually relate the error rate to the bleaching rate and calculate the amount of time that we get one molecule based on the probability of getting one molecule and relate that to the bleaching rate. Now we've got an integral, and we have to optimize an integral. And anyone who's taken upper division mechanics should know what it means to optimize an integral. If you if you haven't taken upper division mechanics yet, don't worry. This is not going to be on the test, but I'm just showing this as an example of places where we got to use our physics bag of tricks to solve problems in imaging. And so we were able to predict acquisition schemes, modeling the number of molecules over time and the activation probability versus time. And we could even ask, all right, well, how do we know we've actually maximized? In upper division mechanics, you learn how to do calculus of variations, set the derivative of an integral to 0. But that just means saying the derivative assigned to 0 could give you a maximum, it could give you a minimum. And we want to maximize the single molecule images and minimize the 0 molecule, the 3 molecule. That was easy enough. The 2 molecule case was special. And we were able to do a lot of calculus on that and show that small error rates, we minimize the 2 molecule error rate. At larger error rates, we are actually maximizing it. And that may sound like a bad thing, but I want to, deviations from the ideal scheme would decrease the 2 molecule error rate. So what we know is that if we deviate from the ideal scheme, we're actually going to be reducing the rate of errors that is most challenging to get rid of. So we've got a scheme where we can select the 2 molecule error rate. Now you may be saying, well, I want it low. Yeah, of course, but 0 is never a possibility. You can never make your error rate 0 in any realistic process. So we can decide how low we want it. And then we can know that if we wind up deviating from that scheme, we will actually decrease it. We'll increase the 3 molecule error rate, but 3 molecule images are really weird images that are going to look out of place. Your image analysis algorithm will be able to reject them immediately. 2 molecule images are challenging, and we've got things set up so that they're at a predictable rate, and if anything goes wrong, we'll actually decrease the number. So it was fun to prove that. I'm going to skip the single wavelength schemes. I want to show you a project I've worked on with my friend at City of Hope. She spends a lot of time studying proteins on cell membranes. Proteins on cell membranes are probably, in some sense, half of biology, or at least they're half of biology for anything multicellular, because it's how multicellular organisms have their cells interact with each other, they interact with their environment. So it's arguably half of biology. She wants to know, okay, how are these molecules arranged? It turns out that they might be clustering on multiple scales, forming 2 molecule clusters and then clusters of clusters or domains of 2 molecules. You might be asking, what's the difference between a cluster and a domain? basically we need a different word to describe two things that are very, very close to each other and a bunch of things that are in each other's vicinity. So we chose these two words so we know what we're talking about. Now, if we have very, very, very high resolution, then we can just look and see this. But in fact, even though we're beating the diffraction limit, we still have a bunch of error. So if these were the actual positions, these would be the localizations, because we wind up analyzing each molecule more than once on average. Looking at this, you could tell right away what's going on. Very clear pairs, and you could definitely see some big voids and some regions where pairs are kind of close to each other. Here, the pairing is by no means obvious to the eye. The domains, yeah, are more noticeable, but what exactly is the size of the domains? So we do statistical analysis on them, and we calculate a correlation function, basically the probability, how far away from this one do I have to look so that I see, what's the probability if I'm looking at this point and looking at something a given distance away from it, that I'll see something else. And looking at the drop-offs it's not actually a probability, but it's proportional to it. Looking at the drop-offs in these correlation functions tells you something about the statistical distributions. In particular, we can look at experimental data and then set up a whole bunch of simulations. And we can never actually make a simulation that perfectly matches the data, especially at short ranges, which tells us that the short range interactions are very complicated. But we can get almost perfect matching at long ranges, which tells us that we do understand the long range interactions. We don't yet understand the short range interactions. And what we're looking at is we're looking at molecules called opioid receptors. And we're looking at opioid receptors on pancreatic cancer cells because clinicians had apparently noticed that, okay, pancreatic cancer is in almost all cases a death sentence because pancreatic cancer is usually found late. There are exceptions. There are people whose pancreatic cancer, just by some miracle fluke, is found early and they have good outcomes. There's a Supreme Court justice who had pancreatic cancer more than 10 years ago and is still alive. And I've got some friends who say, oh, she had pancreatic cancer. She's going to die any day now. If she's had pancreatic cancer for 10 years, she's not going to die any day. She got caught lucky. And it's not just rich and powerful people who get caught lucky. I know a guy who knows a guy who works as a landscaper who just happened to have his pancreatic cancer caught early. But if you don't get it caught early, you're going to die. Now, the thing is that even though you're going to die, you could last a few months. And doctors have anecdotally noticed that people who are taking large amounts of pain medication seem to live longer. Now, there's any number of reasons why pain medication might prolong life in certain types of cancers. It doesn't necessarily have to be that the pain medication is doing anything to the tumor. It could be that the pain medication is reducing the person's stress level. Agony and stress are terrible on the body. As soon as you start feeling pain and stress and agony, it messes up everything else in the body. It could be that. But my friend at City of Hope, which is a cancer research hospital, has been studying the possibility that the opioid receptors on the pancreatic cancer cells do something to the cancer itself. And there's some preliminary evidence for that. Now, I'm not going to sit here and say that tomorrow we're going to cure pancreatic cancer by understanding the clustering of opioid receptors on a pancreatic cancer cell. What I am going to say is that we have a technique for understanding long and short range interactions that wouldn't be obvious even with high resolution microscopes. Will that help us to understand pancreatic cancer? Who knows? Will it help us to understand something that involves membrane proteins? Yeah, membrane proteins are half of biology in some sense. And so, okay, I'm exaggerating. Membrane proteins are a very large portion of biology. If we can understand the interactions of membrane proteins, eventually we're going to understand something that matters. Might be cancer, might be coronavirus, might be something in plant biology, you know, who knows, might be something to do with corn, which, hey, given how much of the world relies on corn, or rice, how much of the world relies on rice, who knows, it's going to be something interesting. But we've developed techniques for understanding membrane proteins. So that's what I do with optics. What I do with optics is I do a whole bunch of theory and computer simulations to try to make sense of visualizing things that my professors told me were impossible when I was in college. Any questions? All right, then. I'm going to stop here. On Thursday, you have one last problem set due. I'm going to be grading problem sets tonight and tomorrow. On Thursday, I'm also going to give out the final project assignment. It'll be 2 problems. One project, it'll basically be like your other ZMAX assignments. One of them will require you to use ZMAX to do something with lenses in a open source plate reader. And the other thing will be to study some situation involving multi-layer films. I'll pose a question involving multi-layer films. I'll say, go use the Python code to run some simulations and figure out what's going on here. So Thursday morning, those will be available on Blackboard. I'll show them, discuss them, take questions. You'll have until the end of finals week to work on them. Anything else? All right, have a good day.